So this is part 2 of DNA fingerprinting. DNA has 4 different nitrogenous bases: adenine, thymine, cytosine, and guanine. These base pairs form pairs according to this rule. Adenine pairs will only bind with thymine, and cytosine only binds with guanine. These pairs are complementary. Remember, at CG. So when we're thinking about it, how does it pair up? If you look at ATCGTGC, the base to it, the base pairing is TAGACG. DNA is a double helix. It looks like a spiral staircase, and it's twisted in a shape. And it has a backbone, and it has a rung. The backbone is a phosphate and a sugar. It's the sugar that binds with the nitrogen bases, and all this binds with a very weak hydrogen bond.
That's what our house.
So DNA double helix is just another picture of it looking at a side view, and you can see the hydrogen bonds. There are 2 types of DNA. There is a nuclear, which we both inherit from our mother and father, and it's virtually identical in all cells of an individual's body. However, the mitochondrial DNA is only inherited through our mothers, and it is unlike the DNA in the nucleus. The human genome is the total amount of DNA in a cell. It's contained in both the nucleus and the mitochondria. There are about 3 billion base pairs. They finally had all of this. It took them a while to get all the codes. The purpose of the genome is to code the blueprint of the human body, and it's responsible for telling the body to make proteins and other needed molecules. DNA identification. Most of the human genome is the same in all humans, but there are some variation among us. VNTR and STR are data from DNA fingerprints and can be analyzed for 2 main purposes. One is for tissue matching, which you can collect from a crime scene and then you collect from a suspect. It will either rule them out or it will rule them part of the crime scene. Inheritance matching is usually used to compare family members of DNA for proof of family relationships. Each band in a child's DNA fingerprint must have at least one parent, 50% mom and 50% dad. So this is at the tail end. What does this tell us just from this gel electrophoresis sample? Mom is all blue and dad is all yellow. What does it tell us about the first daughter? She is blue and yellow. So it tells us that mom and dad are the biological parents. And then look at the second daughter. She is only related from one biological parent, and the other is a different dad. So here is the son, one, and he has both blue and yellow. So he is biological parents. And then we come to the second son, and you see that they do not have any of this. So this concludes my webcast, and we will get back with you as we go more in depth. You all have a good evening.